Good morning, students. Myself, Major Komal Katyal, Department of Zoology, GK Gowani Government College, Bhinmar. In continuation on my lecture on blood clotting, today we will discuss the mechanism, that is the part second of blood clotting. In my last lecture, I told you about the various factors. Now, first of all, we will discuss about the extrinsic pathway. Now, the extrinsic pathway actually requires contact with the tissue factors external to the blood. Hence, it is known as extrinsic pathway. कहने का मतलब यह है कि रुधिर के अलावा भी उत्तक के कुछ कारक जिसमें इन्वॉल्व होते हैं, इनकी आवश्यकता होती है, उसको हम बोलते हैं extrinsic pathway. This occurs when there is trauma to the vascular wall and surrounding tissues. The extrinsic system is triggered by the release of tissue factor, which is the thromboplastin from damaged tissue that activates the factor number 7. This system में उत्तक के द्वारा कारक रिलीज होता है जो कारक संख्या 7 फैक्टर नंबर 7 को सक्रिय करता है. The tissue thromboplastin and factor number 7 activate the factor 10. Now the whole process is shown in this slide, that first the tissue trauma, which is the tissue factor, converting the factor number 7 from its inactive form to its active form, and this activated factor 7 is converting the factor 10, that is the glass factor, and converting it into its activated form. So after the activation of factor number 10, that is the Stewart power factor, this Stewart power factor in presence of calcium ion and factor number 5, that is the proaxillerin or the labile factor, there is formation of a complex which is known as prothrombin activator, and this prothrombin activator in presence of calcium ion and phospholipids from platelet, which is known as the PL factor, converts the prothrombin into thrombin, and this thrombin again activates the factor number 5, and the process goes on. The intrinsic pathway. The intrinsic pathway, the initial reaction is the conversion of factor number 12 from its inactive form to its active form, that is the Hagman factor or the glass factor. And this Hagman factor or glass factor can be activated in vitro by exposing the blood to any foreign surface, say for example the glass tube. The activation of in vivo occurs when the blood is exposed to collagen fibers underlying the endothelium in the blood vessels. So there is no involvement of tissue factor. Now the slide shows the intrinsic pathway, the blood trauma, that is the contact with collagen. There is activation of factor 12 in presence of the high molecular kininogen and Prekallikrein, that is the Fitzgerald factor and the Fletcher factor. There is activation of factor 11, which is known as PTA or antihemophilic factor C, and this activated factor 11 in presence of calcium ion activates the factor number 9 and converts the factor number 9 into its activated form. The factor number 9 is the Christmas factor, and there is action on factor number 8 in presence of thrombin. The factor number 8, which is the antihemophilic factor A or antihemophilic globulins, the AHG, comes to its active form and then it stimulates the factor number 10, which is the Stewart power factor, in presence of calcium ion. So here you can see in the cascade that there is presence of activated factor number 8, activated factor number 9, the calcium ion, and all these are involved for converting the factor number 10 to its active form. Now the factor number 10 in presence of the calcium ion and thrombin activates the factor number 5, and there is formation of the prothrombin activator complex. Again, there is requirement of platelet phospholipids, and the prothrombin is converted into thrombin in presence of the calcium ion. If you can remember the extrinsic pathway, again in the extrinsic pathway there is formation of prothrombin activator which converts prothrombin to thrombin. So this is basically the first step if we consider the broad step of blood coagulation. Now the summary, the extrinsic and the intrinsic pathway, and after the formation of the thrombin there is a common pathway, and during this common pathway the thrombin acts as enzyme which converts the fibrinogen, the soluble protein, into fibrin, and this fibrin in presence of Prekallikrein factor, that is the factor number 13, the fibrin stabilizing factor, there is formation of a clot. Initially the clot is a soluble clot. The whole process which we have discussed earlier is summarized in this slide. The prothrombin converted into thrombin in the presence of prothrombin activator, which is either formed from the extrinsic or the intrinsic pathway in presence of calcium ion, forms thrombin. This thrombin converts fibrinogen into fibrinogen monomer, which in presence of calcium ion into fibrin fibers. Again thrombin activated fibrin stabilizing factor, that is the Prekallikrein factor or the factor number 13, and there is formation of cross-linked fibrin fibers. Now the clot retraction. The fully development of clot takes approximately 3 to 6 minutes. The contraction of platelet trapped within the clot shrinks the fibrin meshwork, pulling the edges of the damaged vessels closer together. During the clot retraction, serum is squeezed from the clot. This is the process of clot dissolution. First, the factor number 12 is acting on Prekallikrein, that is the Fletcher factor, converting it into kallikrein in presence of plasmin, which is basically responsible for clot dissolution, which is present in its inactive form. So first the kallikrein converts plasminogen into plasmin, and this plasmin further forms the kallikrein as well as converts the fibrin polymer into fibrin degradation products. Now let us discuss about the various coagulation disorders. The thrombosis. What is thrombosis? It is the abnormal clotting of blood in unbroken vessels. It can lead to heart attack if the thrombosis or the thrombus is trapped in the coronary artery, the artery which supplies blood to the heart. The thrombus is a clot that attaches to the wall of the blood vessels. The embolus, it is the term used for a clot that comes off the wall of a blood vessel and travel in the bloodstream. It means यह एक प्रकार का रुधिर का थक्का है जो सर्कुलेटिंग ब्लड मतलब जो रुधिर प्रवाह के अंदर बह रहा है। The embolism is the blockage of blood flow by an embolus that lodges in the small blood vessels. It means first there is formation of a thrombus which is initially attached to the blood vessel wall, and if it detaches from the blood vessel wall, it is known as embolus, and when this embolus circulate in the blood circulatory system, it can block the flow of blood if it enters the blood vessels with smaller diameter like capillaries. The infarction refers to the cell death that results from embolism, and this information is responsible for most strokes and heart attacks, which I told you earlier on, if there is formation of embolus in the coronary artery. To summarize the whole process of the blood coagulation, we can say in the stage 1 there is formation of thromboplastin, which in presence of calcium ion converts prothrombin into thrombin, that is the stage 2. And the third one, the fibrinogen is converted into fibrin, initially the soft clot, which later on is converted to hard clot. Now there are various factors which are responsible for affecting the clotting of the blood. बहुत सारे ऐसे कारक हैं जो रक्त स्कंदन या रुधिर के थक्का जमना को प्रभावित करते हैं। उसमें से सबसे पहले है विटामिन के, which is also known as the coagulation vitamin, which is required for synthesis of various factors, which I have told you earlier on that it requires for the synthesis of factor 2, 7, 10 and 9. There is presence of coumarin. Coumarin are derivatives of dicumarol and warfarin, which are effective anticoagulants. Heparin is a substance which prevents the clotting of blood generally inside our body when there is no wound. Calcium, which we have discussed earlier on, calcium ion is required for conversion of prothrombin into thrombin. Then there is process of defibrillation, and this may be prevented by removal of fibrin by certain chemicals like magnesium sulfate. Hirudin is very popular anticoagulant which is generally present in leeches, and whenever the leech sucks the blood of its host, it allows continuous flow of the blood, preventing the clotting of the blood. Apart from it, whenever a mosquito sucks the blood of human being, it also secretes certain anticoagulant substance. We can call it the anticoagulant, which prevents the coagulation of blood. Earlier we have also discussed the process of fibrinolysis, where the plasminogen is converted into plasmin, and this plasmin is converting the fibrin clot into soluble product. And there are certain important factors which activates the plasminogen to plasmin conversion, include the fibrinokinase present in the many tissues, urokinase which is present in the urine, staphylokinase present in certain bacteria, and streptokinase which is present in human fetal cells. The bleeding disorders are the thrombocytopenia, when there is less number of blood platelets, and hemophilia is the hereditary bleeding disorder. So this finishes my lecture on blood clotting. Thank you.